Hello, uh, my name is Chuan Ching. I'm from the physics department of Arizona State University. I'm very happy today to talk about research. Um, this is my contact information, so feel free to contact me if you would like to discuss in more details. And also, I'm providing here a lab tour video made by my students. This is a 15-minute video that tells you how things are made in my lab and how their daily experiments looks like. My research is at the intersect between nanoelectronics and biology. We try to build different nanoelectronic sensors and modulators to investigate biological systems at different levels. For example, we're interested in building single molecule sensors, trying to detect DNA, RNA, and protein molecules at single molecule level. We're also interested in building interface to single live cells such that we can manipulate or modulate the cell signaling pathways to guide their behaviors, change their fate. We're interested in also by understanding how things are coupled to live cells to build interfaces to tissues such that they are more biocompatible. So here I'm going to talk about a few examples of projects that are going on in my lab to give you a sense of the questions we ask as well as their applications. So the first thing I would like to talk about is a solid-state nanopore sequencing platform. The fabrication of this device was actually quite complicated. So you can take a look at the lab tour video, which gives you a more detailed description of how things are made in there. But the structure of the device looks like this. So we construct a small gap. Take a look at here. So this is the small gap over here, where the molecules, it matches the diameter of the molecule and the molecule is forced to go through it. This is the translocation event we referred to. As the molecule translocate, we have a pair of electrodes right next to the electrode, right next to the molecule, such that we can read the tunneling signals across the molecule as it goes through that pore. So we have the ionic current that's blocked by the molecule, telling us that event, as well as the tunneling current across the molecule that tells us structural information about the molecule. In addition, the metal gap also enhances the Raman scattering process. In other words, if you shine a laser on the molecule, you can detect a Raman spectrum that's enhanced by an 8 to 10 orders of magnitude because of the metal structure over here. So in other words, we can actually use multiple parameters to identify with accuracy the chemical information of the molecule as it translocates. So here are the movies that shows the DNA are enriched inside the channel and that it's been stretched and then moved into the nanopore. Each of these peaks, there are correlated signals from the ionic channel as well as the tunneling channel tell us the individual single molecule events. So we hope that we can develop this platform into a single molecule RNA DNA sequencing platform to address many of the limits of the existing technologies, such as to identify directly the modifications of DNA or RNA molecules, because we now have different ways to characterize the molecule at the same time. Now, the second project I want to talk about is how to understand cells sense and respond to external electric field. You probably heard this on the news where people try to apply electric field or inject current into their heads using these simple devices, trying to enhance their memory or cognition performance. This is not totally scientific fiction because there are some evidence that shows other than your neurons and the muscles, which are known to be electrogenic, many types of cells actually respond to electric field. For example, your epithelial cells on your skin, in the wound healing process, they respond to external field. The stem cells, as they migrate and differentiate in the body, they also respond to electric field. However, The mechanism of how cells sense and respond to electric field is actually quite complicated, and there is no clear mechanism that can explain all the response. So if we look at the cell membrane, actually, a lot of things is going on. Many of the sensing process started at the cell membrane. Basically, the sense started with the binding and unbinding of different components to the proteins at the membrane. And the protein respond by changing the conformation and then trigger the downstream signaling pathways. In this whole process, this is a very jammed region where a lot of charges and dipoles interact and they change their positions. Therefore, when you apply an external electric field to this region, then it's very likely that you're going to shift the activation energy of this enzymatic process, which would then tune how the downstream signaling pathway network would behave. This is an example we recently showed that you can actually electrostatically bias or shift the ERK signaling pathway to inhibit that pathway network or activate the pathway by tuning the waveform and timing of the electric field you apply to the cells. And the ERK pathway is actually one of the essential pathways that regulates gene expression, plays an essential role in cell migration, cell proliferation, cell differentiation, and cell apoptosis. So the idea is that if we understand how to tweak the electric field to target the specific components of the cell membrane, then we have another dimension of freedom to program the cell and guide the differentiation or the fate of the cell to a certain direction that we want it. The last project I want to mention briefly is how mechanically you can actually stimulate the cell. So we built this tunnel topography features on the surface, and we noticed that it actually regulates the fibronectin signaling pathway on the cells. In other words, how the cells actually construct their cytoskeleton structure, how they migrate and move on the cells, can be regulated by a simple nanotopographical features. And what's surprising here is that the feature itself is only 100 nanometers, while the triggered response from the cells are a micro size. And these responses are spatially confined within those features. So if we can understand how molecularly these two interact with each other and how they actually construct this spatial and temporal wave-like propagation behavior, then we may be able to design the surface to guide the cells to do things that we want them to do. So to summary, I think there are many biophysical questions in the interaction between artificial devices and environment and live cells and tissues, and we hope that we can understand this coupling and also then find their biomedical applications. Okay, so that's all. Thank you.